Talk to us about the difference between like an FMT product and then a fermented product, and why you've gone big in the fermented arena, so to speak. Or
I think, this is my feeling. I think in the future, I think the FMT products will prove concept for sure, and I think there is something with that full spectrum of. There's an interplay that still don't think is understood. Right? There's fungi and there's all sorts of other stuff that's in there that I don't think it's well understood. Right? And there's we've seen some analytical data on. You need certain strains for other strains to propagate. There's an interplay between certain strains. That certain strains produce metabolites that other strains need. There's a lot of that stuff. And so I think FMT, will be the first in class treatment for a lot of these things. And I think that's...
So a donor-derived product. The starting material is stool. It's been rigorously screened from healthy people. And you're trying to retain the ecosystems as best as possible, right? And you mentioned the various different elements there. Do you think a fermented product, can you achieve the same kind of complexity, or you can achieve enough?
Well, yeah, so I think there's a number of different ways to think about that. I think there's different ways you can go. One of the nice things about being a CDMO, and I want to make sure I don't talk about any one specific product, but
no problem.
We get to see lots of different approaches. We even get to people come to us and say, hey, we're thinking about this thing. How do you think about it? How would you manufacture that? But there's other ways to approach this, which is things like, bringing, consortia together, bring 50 different strains together, either make 50 different drug substances, which would be difficult from a COGS perspective, or could you co-culture them together? Is there other ways? Could you do something where you have a background of FMT, use that as a scaffold or an excipient, something like that,
right? And
then focus on 2 or 3 bugs,
right?
I think there's a lot of different directions it can still go in,
right?
And for me, it's just I think there's some data out there that says that certain strains of bacteria supplement with a frontline oncology treatment improves the responsiveness. To me, it's just a matter of time.
I saw some data recently, still unpublished. I can't go into all the detail, but FMT or IMT, intestinal microbiota transfer, however you want to describe it, as an adjuvant treatment in patients who are receiving immunotherapy. And the data was incredible. I got really excited, both because I'm running a microbiome company, but also with my clinician and sort of just human hats on. I was like, oh my goodness, if we can achieve like the disappearance of cancer cells in a significant proportion of patients and also shift non-responders to responders to immunotherapy, that has such massive implications globally. Yeah. Quality of life, lifespan, all this kind of stuff.
That's a great example, because it's not that long ago people thought it was a genetic disposition that meant people were responders. So to think that it's actually the microbiome, and by being able to modulate that, you can improve it. To me, it's just
wow.
It's the same. I think if you think about how long ago was it? There was this sort of cesarean births typically have higher levels of asthma,
right,
and other kind of skin conditions, and sort of was like, well, people didn't really understand why that was. And now it's pretty clear. It's because the microbiome is populated during the birth process.
That's it.
And, nature has a way of just knowing and sort of being there for a reason. And I just, for me, in the next 10 years, I think it's a really exciting time to be part of the microbiome.
Right. Yeah. On the analytical side then, and I take the point that we can't go into any specific, client customer detail. Sure. I wouldn't push you on that at all, but what are you doing at Aranta to innovate in the analytical side? And do you see that as being like a bedrock foundation of your offering and your business? Like, how do you compete with other CDMOs to make yourselves like, the one that people go to?
Yeah, no, we from really early on, so we obviously acquired Cap Design. So Aaron Cowley, who's our CSO, is still with us in the organization. He did some brilliant work on how do you grow bugs and keep them alive through the manufacturing process. I think if he was here, I could say this, I think there was a little weakness on the analytical side, and actually we brought this guy, Jeff Heiser, on board, who's just like unbelievable.
He was at Boston Analytical, right? Yeah, I've heard him talk a few times.
And his passion for analytical aspects of microbiome is just brilliant. He's constantly thinking about how does we innovate further on analytical. But for us, one of the early challenges, it's a really simple one, was a lot of the products are consortia. So you're combining 9, 10 different strains into a single drug product capsule. Then how do you make sure you've got the right quantities and all that stuff? And
so do you need to have the same amount by gram weight of every strain in the capsule?
Yeah.
Wow.
And then if you think about that, as you think about for a commercial process, at some point you're going to have to do process validation and show that every time you make it, it's got the same.
So can we just pause there? When you move through development through to commercial, from the manufacturing and analytical perspective, generally speaking, what changes?
I guess the precision of the analytical has to be tighter and tighter and tighter as you go through. We actually still see in a little bit in microbiome, people want to do a lot more on the analytical side earlier. But in general speaking, as you get closer to phase 3 and validation and process characterization, you want to make sure that you really have a strong understanding of what you're doing in the manufacturing process and what the product, all of the the potency, safety, all those attributes are right.
Because I guess when people were watching the vaccines being developed, it was all about phase 1 to phase 3 or phase 2 to phase 3 and getting the data. But I think a lot of people don't realize just what goes into all the rest of the elements. Supply chain.
It's incredible.
Analytical.
Yep.
And you're at Aranta, are you offering an end-to-end solution for your clients?
Lab to commercial. And I think for microbiome companies, what's important is obviously they want to get through phase 1 quickly. Time is money. Everything before phase 2 has a negative net present value. So money's, they want to really go as quickly as possible. But I think what's important to their investors is they have somewhere they can actually manufacture in the future. So the fact that we invested ahead of time
in the capacity.
Yeah, we have phase 3, we've got commercial ready facility, but we have a commercial ready quality management system that was designed that we could take people through to that in the future. Now, most products are in phase 1, phase 2. We don't need that. What we see for microbiome companies increasingly is they kind of go through to phase 2. There's a growing awareness of how important CMC is. There's a lot of people in the sort of preclinical phase 1 who don't fully understand why. We're just to be clear, Aranta is not the cheapest option. We're in the middle of Massachusetts. We talked about it's a $150 million facility that we've got. We're not the cheapest. So we have to spend a lot of time sort of getting people to understand why do we need all these controls? Because we've got to make sure that the product is pure. It doesn't have contamination from any other products and processes. And I think one of the things is probiotics typically are made in multi-use facilities, but they don't have the same controls.
I don't know much about the probiotics industry in terms of who manufactures them and how they manufacture them. My understanding is that, as you say, because it's not a drug product, then the principles of GMP don't apply, although they might embody some quality management.
They would say, they call it food grade GMP.
But it's not, how can you be food grade in GMP?
Well, so it's still, but it's not pharma grade GMP. So they would do less analytical. They would probably do less on the containment.
Just for the listeners, GMP means good manufacturing practice. Yeah. And you're probably better at describing it than I am, but I'll have a shot, having, had some regulatory interactions quite recently. The way I think about it is it's a mindset.
Yeah.
It's a mindset relating to quality, consistency, training, quality management system, making sure that you're producing the right product same way every single time. And if you don't, you've got to capture that and log it. And ultimately, what it boils down to is patient safety.
Yep.
Making sure that these products are safe for patients every single time.
Yep.
Anything to add as the GMP expert?
I'm lucky. I've got much smarter people around me. But
me too, man.
No, I think that's pretty much it. I think for me, GMP is generally common sense, and I think if you're getting your medicine made somewhere, you want to know, like,
right,
the piece of equipment it was made on wasn't previously made, didn't have a steroid on there that is going to carry over into the product. I think for probiotics, by design, they're just not to the same level. And I think it's funny, Aaron, we asked him about probiotics at the very start, tell us the difference. And he said, well, first of all, if you buy probiotics from the supermarket, you waste your money, because although it's got like 1 billion CFU, all those CFUs are dead. They're not viable.
Wow.
So you're wasting your money on those £50, £50 things.
And by the way, having been to America a couple of times this year and gone to the supermarkets, which are they're huge.
Yep.
The number of probiotics for sale blew my mind.
Yeah.
And it was a very, dare I say, I hope I'm allowed to say this, American way of marketing. We have the most cells in this product, right? They didn't say anything about what the bug was or whether they were viable.
And they're sitting on the shelf. They're not exactly. There's no. Yeah. But even we still find ourselves having to describe that to people, and when we bring employees on,
there's a big difference, right?
It's huge.
Yeah,
it's huge, and I think that's the trick for microbiome is for fermented products certainly, and in fact even for yours, to make sure that the cells are one are alive at the end, and that they hit the right part of the GI tract where they can propagate and grow again. And
right.
It's not easy, and I think sometimes the industry thinks some of these things are easy, but it's not. I mean, we're running the facility for microbiome the same way as we would run it for mRNA, and the same way we'd run it for a viral vector. Which these are types of products that would command significant prices in the commercial market, and microbiome's no different. We have the same controls. In fact, because of the manufacturing complexity and the randomness of when stuff needs to be harvested, in many ways it's actually more labor intensive.